So now we look at this anatomy of the neuron. This is a schematic diagram, right? Just note that, of course, I mean our real neuron do not actually look like this. This is just like schematic rendition, all right? But all the elements are here, right? Dendrite, cell body, also called as the soma, SOMA, and then we have the nucleus, we have axon, Hulok, right? Exxon Hulok, just take note, right? It is the point, all right, the location where our action potentials start, right? This is where it starts, right? And then we have all these myelin sheaths, right? These are actually adipose tissue, right? These are fatty tissue. And then you will actually have all the action potential being regenerated at all the junctions of our this myelin sheath. So in that case, this really speeds up the neurotransmission, right? It ensures that everything gets transmitted, as fast as like 3 milliseconds, right? Yeah, below 3 milliseconds. Right. So these are the nodes of Ranvier. That's what I'm talking about. All these junctures, right, between all these myelin sheath, right, myelin Schwann cells. Yeah. And then later on, all these signals will get propagated until they reach our this terminal buttons, right? That's our textbook now, right? But usually they're just called exon terminals, right? And then collateral branches. So collateral just means that the same kind of signals, right, with the same frequency is actually sent to both branches, right? So that's a purpose of this biological design, right? So we have the same degree, the same magnitude of neurotransmission, right? And then all these axons they get split up into all these mini branches. Yeah, that's how it's like. And then, all right. So before I talk about neurotransmitters, so neurotransmitters basically they are found where, right? Dendrites, yeah, dendrites are receiving all these so-called neurotransmitters, and then at our terminal buttons, right, axon terminals, right, we have all the neural transmitters getting transmitted across the synapses, right, to another, dendrites. Yeah, so that's how it works, right? All right, now I will talk about all these different substances. So in general term, they should be called neurochemicals, right? So just now, if you guys remember, right, glutamate, GABA, right, this strictly speaking, these are our neurotransmitters, right? Neurotransmitters is actually used specifically to refer to neurochemicals that have a direct impact, right? They diffuse directly across the synapse, and they have a direct action on the postsynaptic receptors, right? The postsynaptic receptors will accept them, and then that will incur a cascade of biochemical reactions, right, in the receiving cell. And now when we look at dopamine, serotonin, right, these are what we call catecholamines, right? These are neuromodulators, more specifically speaking, and then their action can be, is a bit slower, and then they can, bind to some postsynaptic receptor that does not really initiate a direct cascade of biochemical reactions. They can bind to one particular kind of receptor, and that receptor will actually start off a cascade of events that affect another neighboring receptor, right? So that's like a more indirect kind of biochemical action, right? So dopamine, serotonin, they behave in that way, right? And ACH, right? All right. ACH, this is more like another. I couldn't really recall what we actually call them at the top of my mind, right? But it is the first neurochemical that's being found, and it is highly involved in learning and memory, yeah, memory consolidation, right? And endorphin and enkephalins, right? This for pain relief, right? Yeah. So we heard about this endorphin rush, right? If you kind of did a lot of running, and then yeah, some people experience it, right? Yeah, not everybody do that. Okay, so before I go on, just want to remind everybody. Okay, so serotonin is is highly associated with the manufacturing of SSRIs, right? So that is like, what we call selective serotonergic reuptake inhibitor. So that is the category of medical drug that is used to treat depression, right? Major manic depression, right? So the typical depression. So we try to, as much as possible, right? The psychiatrist, the doctor would try to increase the concentration of serotonin in your synapses. So that's the bare bone function of it, right? So it is kind of believed, like, because we have so many serotonergic pathways, right, in our central nervous system, right, our brain, our spinal cord. So it's kind of like, if you can somehow artificially increase the serotonin concentration, and then, smoothen the passage of serotonin in your brain to your spinal cord, it is kind of believed, right, that that may alleviate certain depressive symptoms, right? So we have drug called like fluoxetine. Yeah, I'm sure some of you heard of it. Fluoxetine, right? So that is a typical SSRI. Yeah. However, this does not really work for everybody, right? Just to remind everybody, yeah, it does have certain side effects, right? And SSRIs are usually very expensive, right? Dopamine is, possibly, in my opinion, the most famous neurochemical, right? Yeah, because it's involving attention, arousal, and all kinds of activity, right? That requires concentration, that requires you to be in a high mood, right? Something like that. And then when you kind of abuse drugs, like cocaine, for instance, right? Cannabis, cocaine, marijuana, methamphetamine, right? Yeah. So all these things, right? If you've been to America, right, you'll see a lot of people abusing such things. So such people, what are they doing? They are consuming all those leisure drugs, right? All these leisure drugs. They're trying to, upregulate the dopamine in the brain. Simple terms, right? Yeah. So it's um, yeah. I mean, even though those drugs initially, like, I mean, cocaine, it's supposed to be like pain relief, yeah, but people are just abusing it, right, to the extent that it has such a bad reputation right now, right? It's very, very unfortunate, right? And then GABA is the primary inhibitor, the primary inhibitory neurochemical in our brain, right? So when we talk about inhibition, right, later on when we go to the action potential part, right, inhibition is associated with hyperpolarization, right? Just I will show you guys the figure. And then when it comes to glutamate, right, glutamate brings about excitation. So that is involved with the generation of action potential, right? So this is from one of my previous lecture, right? This is PY 2Y01, right? This is just to benefit everybody, right? I really want to showcase a distinction between action potential and graded potential, right? So action potential, we know what it is, right? We know what it is, how it looks like, right? It's just your upward spike, and then it goes down, and then there's this dip, right? And then everything starts at negative 70 millivolt, right? That's the so-called baseline, and that is what we call the so-called resting potential, right? Just do take note that this does not really apply to every single cell in our body, every single neuron, right? This is taken like, a statistical average, right? So when everything goes up, right, everything depolarize, all right. So from negative 70 all the way to like 40 millivolt, right? That is like half. We call it the rising phase of the action potential, right? And that is brought about by sodium cations, right? So if I'm not mistaken, right? So initially, right? there is a difference, right, in the concentration of sodium and potassium ions, right? So there's always a bit more sodium cations outside the cell, right, than inside, right? So to get the rising phase, you need to get the inflow of the sodium ions, right? And then when it goes back to the resting potential, or the reverse happens, right? There is this outflow of extra this potassium cations, right, to it out to it out of the cell, right, out of the membrane, and that really brings everything back to the resting potential, right? I mean, that's just a very, very simple way of putting it, right? But we also need to consider, like, other kinds of ions as well. There's also calcium ions, there's also chloride ions, right? But for the sake of this module, right, we don't really need to go into that that detail, right? So that is really for PY2Y01 later, right? So that is the action potential, right? So, but what is the graded potential? So when you look at the greater potential, right, this so-called EPSP, right, excitatory postsynaptic potential, and then we have another thing called IPSP, inhibitory postsynaptic potential, right? You don't really have to know what exactly they are because you guys will learn about it, right, in PY2Y01. But all you need to know is that, when you look at this EPSP, right, it is graded, meaning that, it does not lead to this a sharp spike. So it's just a slight increase, right, in the millivolt, right, in the voltage. So from negative 70, right, it goes to negative 65, and then after that, it falls back to the resting potential. So this is what we call a graded potential, right? And this is like a graded excitatory potential, right? It can also be a graded inhibitory potential like this. When you look at this IPSP, all right, so this is a typical, inhibitory graded potential. Yeah. So from negative 70, instead of going more positive, it becomes more negative, right, to negative 75, right? So this is how it's like, yeah. In simple terms, right, you have to know about all these differences, in terms of differences in all this voltage, right, potential differences, right, in other words. And then what this graph is actually showing is that there's this neural integration mechanism. Our brain cells can actually do math, right? Yeah. When you get excitatory potentials and inhibitory potential together, everything can actually sum to baseline, which is really fantastic.